All right, so I'm going to show you a picture, and I want you to think about it and pick what you think is the best answer to this question, okay? And take about a minute to do it. I'm going to start the clock right now. Okay, so about 15 seconds. 5 seconds. 4, 3, 2, 1. All right, let's see how we did. You guys are too smart. Okay, so pretty much everybody said, this is an amphipathic molecule. That's right. Okay. Now, does anybody know what is that molecule? Anybody know what it is? Bonus. What is it? Not ATP. No, it's called phosphatidylcholine. Okay, phosphatidylcholine. So phosphatidylcholine is one of the most common components of animal plasma membranes. Okay, it's a very common component. So it's an amphipathic molecule. So here's the 3-carbon backbone right here. Right, here's the 3-carbon backbone. Here's the phosphate head group. It actually has an additional functional group on it that makes it choline. Okay, so that's why it's called choline. If this was something else, it would be a different kind of phospholipid. And then we've got these fatty acid chains here, one of which has an unsaturated site, but it's actually it's not a trans fat, right? It's a cis carbon double bond. So that's a type of fat that we often see biologically, as opposed to the trans fats that occur in hydrogenated vegetable oils that we talked about, right? So this is not something you would expect to see in hydrogenated vegetable oil. It's not going to end up in the cell wall, right? Because it's in the plasma membrane. The cell wall is found in plant cells where we find a lot of cellulose, and the synthesis does not involve hydrolysis, but actually dehydrogenation. Help me out. Thank you. And it's also not a type of triacylglycerol, because in triacylglycerol, we would actually have a third fatty acid chain here, not this phosphate head group. Okay, so that's that. And I think you guys all knew that because everybody got it right, so that's terrific. All right, so let's now talk a little bit more about osmosis. So this is the process of diffusion across a selectively permeable membrane. So water will diffuse across a membrane from a region of lower concentration of solute to a region of higher solute concentration, as we just talked about. And I won't belabor this point too much because I think it's sort of something that a process that we're all familiar with, that if we have a situation where we have a semi-permeable membrane in which we have a solution with higher solute concentration on one side relative to the other, over time, water will tend to diffuse across the membrane such that, that because initially there's a lower concentration of solute over here, over time, those concentrations will equalize. And so we're going to see a lower level of water on this side if this membrane is only permeable to water and not to these solute molecules. And this kind of shows a diagram of how this works, where in this case, they're showing you an example where we have solutes that have water molecules very highly ordered on the surface of those molecules. So that tends to affect the way that these water molecules are behaving. And so this leads to this business about water balance of cells. And these are cells without walls, by the way, so these would be animal cells. And there's a few terms that you should be familiar with. So the term tonicity is the ability of solution to cause a cell to gain or lose water. If we have an isotonic solution, that means that the solute concentration is the same inside and outside of the cell. So there's no net movement of water across the selectively permeable membrane of the cell. But if we have a hypertonic solution, this means that there's actually a higher solute concentration outside of the cell than inside the cell. And over time, the cell will tend to lose water. And conversely, if we have a hypotonic solution, we have a lower solute concentration outside of the cell than inside. And over time, the cell will tend to gain water. And your book has a very nice drawing that kind of illustrates this process very graphically, both for animal cells that have the plasma membrane on the exterior, that have only a plasma membrane on the exterior, no cell wall, versus plant cells that have the plasma membrane, but they also have this very thick cell wall. And so what we see is that in a hypotonic solution, so again, that means that there tends to be a lower concentration of solutes like salts outside compared to inside. Over time, water flows into the cell. In fact, we can use this as a way to break open animal cells. By just incubating them in a hypotonic solution, many of these cells will just lyse. They will break for this reason. Just physical pressure of the water that goes into the cell breaks the membrane open. But interestingly, in plant cells, that's not true. Actually, plant cells are happiest in this kind of environment, a hypotonic solution, where water will tend to go into the cell, but because of the cell wall, these cells are very rigid. And so they develop what's called turgor pressure, where the cell wall actually prevents too much water from going to the cell. It goes into a point, and then the cells are very firm, and then more water, it's difficult for more water to enter the cell. This is why at the supermarket they spray vegetables with water. They're not spraying with salts, right? They're spraying with water, not salt solutions, for this reason. Okay? It keeps them looking very fresh. Isotonic solutions, as we discussed, solution salt concentrations inside and outside are about the same, so the water flow across the membrane is about the same. But in a hypertonic solution, cells tend to shrivel up, right? They tend to shrivel up because they tend to lose their water over time to the external solution. Okay, so that's also not so great for cells. And of course, cells have lots of ways to control this kind of thing because they're not always in an isotonic environment, right? So they find ways to do what's called osmoregulation. They control the ability of water molecules to move across cells. And part of the way that they do that is with transport proteins. So, for example, there are transport proteins called aquaporins that, like the name suggests, are pores. They're pores that are made by proteins that are transmembrane proteins that control the ability of water molecules to get inside or outside of the cell. Okay, so they facilitate the passage of water. A couple other things here. So some transport proteins are called channel proteins, and they can have a hydrophilic channel on the interior that allow ions or other kinds of molecules to go across. So on the very first slide, it's actually on the very first page of the textbook chapter, is a picture of a potassium channel, and where you're looking down at the actual you're looking at the surface of the membrane. And there's a channel going through the center of that potassium channel that allows potassium ions, very selectively, just potassium ions, not other kinds of ions, to go across that channel. Okay, so these can be really very highly selective kinds of protein channels. Okay, so facilitated diffusion. So this means passive transport, but it's aided by proteins. So in facilitated diffusion, we have transport proteins that can help increase the rate of movement across the plasma membrane. Okay, so these, like the name says, they kind of provide these corridors or pathways that allow a specific kind of molecule or ion to go across the membrane. These include aquaporins, as we just said, but they also include channels for ions that can open or close in response to a different kind of various kinds of stimuli. And these are called gated channels. And this is, again, a very schematic drawing of what these do. So when we have a channel protein like this, we have a situation where these molecules can literally travel across a little tunnel that's created in the membrane by this protein, like the potassium channel example. And down here, this is showing you what the book defines as a different class of these transport proteins, namely a carrier protein, whose job is to bind to a particular kind of molecule and then, often through a change in the conformation of that transport protein, release it on the other side of the membrane. So this is showing you an example of that kind of process. And in passive transport, this is just occurring without requiring energy, okay, without requiring ATP hydrolysis. Now, in contrast to passive transport, we also have active transport. And in this case, we have a situation where energy is required to move various kinds of molecules against their concentration gradient. Right? So facilitated diffusion, as we just discussed, is still a passive process because the solute is moving down its concentration gradient. But in some cases, we have transport proteins that actually can move solutes against the concentration gradient. And this is active transport. So it requires energy in the form of ATP, and it's also performed by very specific kinds of proteins that are embedded in the membrane. Right. Okay. So this is a really nice example of active transport, and this is a super important process because this is a process by which the cell maintains not only the balance of salts across the membrane, so between the interior and the exterior of the cell, but it also, as we'll see, maintains a voltage across the cell membrane as well. Okay. And so this is a sodium-potassium pump that functions to remove sodium ions from the inside of the cell and release them outside of the cell, and vice versa to bind to potassium ions that are outside of the cell and release them inside. So let's just go through how this works. And so the idea here is that we have an integral membrane protein that is capable of binding, has binding sites for potassium ions. And what various experiments have shown is that typically for each cycle of pumping that goes on in this active transport process, there are 3 potassium ions per transport protein that are bound and released across the membrane. Yeah. He's asking, is the selective permeability of the cell membrane similar to the blood-brain barrier? Well, I think the gross principles are the same, right? That you have a barrier that has selective permeability to molecules, but the details, of course, are in terms of the molecules involved in creating that barrier and what's allowed across and how that works are we're talking about sort of 2 separate things, right? But the principle is similar. Okay. And so the idea is that we have these 3 sodium ions that will bind to this transport protein, and when they bind, that actually triggers an event called a phosphorylation, in which, and we'll talk a lot more about ATP next week, but what you need to know for now is that this means that a molecule of ATP that's inside the cell will donate one of its phosphate groups to a specific place on this transport protein. And when it does that, it tends to, so now this protein contains an extra phosphate group, and that might sound like a very small chemical change, but what crystallography, X-ray crystallography has shown is that this kind of phosphorylation event can actually trigger a conformational change in a protein like this. And when that happens, it actually allows this transport protein to now release these sodium ions on the other side of the plasma membrane, in this case on the exterior side of the membrane. So those 3 ions are released, and now when they're released, that exposes binding sites that are highly selective for potassium ions. So these now will bind to this transport protein, and that binding event in turn triggers hydrolysis of this bond, so that this phosphate group is actually now released off of the transport protein. When it gets released, that kind of resets the system. So this protein now gets reset in its original configuration, and when that happens, now the binding affinity for these potassium ions is a lot lower, and so they tend to be released into the interior of the cell, and then when that happens, it's now fully reset, and it's capable of now binding to 3 sodium ions, and the whole cycle can happen again. So this is an energy requiring process because we're burning up 1 molecule of ATP. When I say burning up, that's a very colloquial term, of course, right? But we're basically metabolizing 1 molecule of ATP for each cycle. Yeah. Yeah, so that's a great question. Why does this thing, how can it tell the difference between sodium and potassium? So those ions have different sizes, right? They also have different coordination geometries, so they have some interesting chemical differences. And it turns out these types of proteins take advantage of those seemingly small but very important chemical differences between these ions to create very specific interactions that allow them to recognize one ion and not a different ion. Okay, and again, how do we know that? We know that largely from X-ray crystallography, where people have been able to trap these kinds of proteins in different states of this cycle, where they can see the types of interactions that occur when particular ions are bound to them. Okay. The phosphate, right, so this hydrolysis event, so all of this occurs through these changes that are occurring in the protein structure. So, this is a very schematic drawing. It doesn't really do justice to this, but you have to remember this is a whole, it's a 3-dimensional polypeptide structure that where the conformation of the protein actually affects the stability of a bond like this, okay? Right, so ion pumps are very important for maintaining what we call the membrane potential. So the inside of the cell, it tends to be negatively charged compared to the exterior. And you probably noticed that if you're pumping out 3 positively charged ions for every 2 positively charged ions that are coming into the cell, that's going to tend to generate a situation where you have negatively charged environments inside of the cell. And these combined forces are collectively called the electrochemical gradient because there's both a gradient of ions across the cell membrane, but there's also a gradient of electrical charge. And that's actually really important because cells can use both properties to drive the transport of other kinds of molecules. Okay, so we have a chemical force, which is the ion's concentration gradient. We also have this electrical force, which is the effect of the membrane potential on the movement of an ion. And we'll look at a couple of examples of this. So an electrogenic pump is a transport protein that generates voltage across a membrane. So this example that we just discussed of the sodium-potassium channel is an electrogenic pump. And it's actually the major type of electrogenic pump in animal cells. In plants and fungi and bacterial cells, the major type of electrogenic pump is a proton pump. So rather than pumping salts and sodium-potassium ions across the membrane, we're actually pumping protons across the membrane. And this next slide just shows an example of this kind of pumping that goes on, where often cells can couple these types of gradients to allow transport of different types of molecules. So what you can see here is that we have a proton pump that's embedded in the membrane that is an energy-requiring pump. So it's burning ATP molecules to pump protons from the interior to the exterior of the cell. When that happens, it helps to create this electrical gradient. It also creates a chemical gradient. And then when these protons are outside of the cell, they can bind to other molecules, in this example to sugar molecules like sucrose, and that allows these sugar molecules to then move across the membrane against their concentration gradient and pulling these protons back into the cell in a favorable direction with respect to the proton concentration. Okay, so in this case, both the concentration of these protons and this electrical gradient are being used to drive the transport of sugar molecules in this direction here. Okay, and then finally, I want to just discuss very briefly bulk transport in cells, right? And so we've been talking about these individual channel transport proteins that operate by interacting with individual molecules, but in some cases, cells actually use a process called exocytosis or endocytosis to create vesicles that can either import things into the cell or release things from the cell. And this is typically done for larger molecules like polysaccharides and big proteins. And of course, this is a process that requires energy. It's an energy-requiring process in the cell. Exocytosis means transport vesicles are migrating to the membrane of the cell, fusing and releasing their contents from the cell. And endocytosis is kind of the opposite. The cell takes in a macromolecule by using a piece of the plasma membrane to pinch off a vesicle that engulfs a macromolecule and pulls it into the cell. And there's a very nice figure in your book that's shown right here that kind of illustrates 3 examples of vesicle transport that are shown here. Phagocytosis, so this sort of means eating, right? So this is the way that cells can actually engulf food molecules and pull them into the cell inside of these vesicles. And this shows an amoeba that's eating a bacterium that way by phagocytosis. We have penocytosis, where cells create these, pinch off these vesicles that are binding to very small molecules. So the word peno means small. And then finally, receptor-mediated endocytosis, in which cells actually have receptors that are localized in certain places on the membrane that bind to particular kinds of molecules and concentrate them there, such that vesicles can then pinch off and engulf them and pull them into the cell. Okay, so study these processes in the book, and have a nice weekend. See you guys Monday.